Assalamualaikum and hello everyone. Welcome to our video assignment for Bio 565, Comparative Biology of Reproduction. Today, we will be analyzing the case of a 32-year-old woman who has been trying to conceive for over a year. She presents with irregular menstrual cycles ranging from 30 to 50 days, inconsistent ovulation patterns, and reduced level of luteinizing hormone surge. Her husband's sperm analysis is completely normal, meaning the reproductive challenge lies on the female side. I will be explaining the physiological process of ovulation, focusing on the hormonal roles of FSH, LH, estrogen, and progesterone. Ovulation is the release of a mature oocyte from the Griffin follicle, and it is controlled by the hypothalamic pituitary ovarian axis, or HPO axis. It begins in the pre-ovulatory phase. The hypothalamus secretes GnRH, gonadotropin-releasing hormone, which stimulates the anterior pituitary to release FSH and LH. FSH acts on the ovarian follicle, stimulating the growth and maturation. As the dominant follicle grows, its granulosa cells convert androstenedione into estradiol, a form of estrogen. In the early follicular phase, this rising estrogen initially exerts negative feedback on GnRH, FSH, and LH, keeping them in check. However, and this is critical, once estradiol reaches a high enough threshold concentration, the feedback switches from negative to positive. This means estrogen now stimulates further GnRH release, which drives a massive sudden surge of LH from the anterior pituitary. This LH surge is the key trigger for ovulation. It activates proteolytic enzymes within the follicle that degrade the follicular wall, forming a weak point called the stigma. The mature oocyte, surrounded by its cumulus oophorous cells, is then expelled through the stigma into the peritoneal cavity, where it is captured by the fimbriae of the fallopian tube. After ovulation, LH continues to act on the remaining follicular cells, transforming them into the corpus luteum, the yellow body. The corpus luteum then secretes progesterone, which thickens the endometrium and prepares the uterus for possible implantation. If fertilization does not occur, the corpus luteum degenerates, progesterone drops, and menstruation follows. In a normal 28-day cycle, the entire process is tightly timed and predictable, but in outpatient, with cycles ranging from 30 to 50 days, something in this hormonal axis is dysregulated, and that is what we'll explore further.
Thank you. I will now evaluate the importance of the LH surge in reproductive function and then walk through the key biological steps of human fertilization. The LH surge is not merely important, it is non-negotiable for ovulation. Without it, even a fully mature Graafian follicle will not rupture. The surge must be sufficient in both magnitude and duration to trigger the cascade of events leading to oocyte release. In our patient, the reduced LH surge means the trigger is too weak. This leads to what we call anovulation, failure to ovulate, or oligovulation, infrequent, unpredictable ovulation. Anovulation is the most frequent cause of female infertility. The disruption in LH signaling also means the corpus luteum may not form properly, reducing the progesterone output and making the endometrium poorly prepared for implantation, even if fertilization somehow occurred. The cause of this reduced LH surge in our patient is slightly hypothalamic pituitary ovarian axis dysregulation, where the signal from the hypothalamus to the pituitary is altered, resulting in insufficient gonadotropin secretion. Factors such as chronic stress, body weight changes, or idiopathic hormonal imbalance can all contribute to this. Now, let me describe the mechanism of human fertilization. Fertilization normally occurs in the ampulla of the fallopian tube. Of the 200 to 300 million sperm deposit in the vagina during intercourse, only about 300 to 500 actually reach the oviduct. Before sperm can fertilize the oocyte, they must first undergo capacitation, a 7-hour conditioning process inside the female reproductive tract. During capacitation, the acrosomal membrane becomes destabilized and membrane permeability to calcium ion increases, which enhance the sperm motility. Fertilization then proceeds in 3 phases. Phase 1. Penetration of the corona radiata. The sperm releases hyaluronidase from its acrosome, an enzyme that digests the hyaluronic acid matrix holding the cumulus cells together. This disperses the corona radiata cells and exposes the zona pellucida beneath. Phase 2. Zona pellucida penetration and acrosome reaction. The sperm binds to the zona pellucida via glycoprotein ZP3 and ZP4, triggering the acrosome reaction, the release of enzymes including the exterase, acrosin, and neuraminidase that lies the zona pellucida. The sperm physically drills through, reaching the oocyte plasma membrane. Phase 3, membrane fusion and zygote formation. The sperm and oocyte plasma membrane fuse. The secondary oocyte, which had been erased in metaphase 2 of meiosis, now completes its second meiotic division, producing the mature ovum and a second polar body. The sperm nucleus decondenses to form the male pronucleus, while the oocyte nucleus becomes the female pronucleus. The true pronuclear fuse in a process called syngamy through fertilization, restoring the diploid number of 46 chromosomes and forming the zygote. To prevent polyspermy, two blocks are activated. The fast block, which is immediate membrane depolarization via sodium ion influx, and the slow block, which is the cortical reaction, calcium ion mediated exocytosis of cortical granules that hardens the zona pellucida and it activates sperm's binding receptor.
I will now analyze how this patient's condition directly impacts fertilization, propose two evidence-based interventions, discuss our strategies, and predict what happens physiologically if fertilization occurs but implantation fails. In a normal cycle, ovulation is predictable. The oocyte is released around day 14 and remain viable for only 12 to 24 hours. Sperm, on the other hand, survive for up to 48 hours in the female tract. This creates a narrow, fertile window of roughly 1 or 2 days. In our fashion, irregular cycles, 30 to 50 days mean ovulation. If it occurs at all, happens at completely unpredictable times. Coupled with a reduced LH surge, ovulation may not occur in many cycles. Even in cycle where some hormonal activity takes place, the surge may be insufficient to produce a fully mature, fertilizable oocyte. Aged or optimally matured oocytes are also more vulnerable to polyspermy and failed pronuclei formation. The consequence? The narrow fertile window is almost impossible to identify, and even intercourse is timed correctly, the oocyte may not be present or viable. This explains why this couple has been unsuccessful despite normal sperm. Intervention 1. Medical ovulation induction. The first intervention is pharmacological ovulation induction. The drug of choice is clomiphene citrate, which works by blocking estrogen receptors in the hypothalamus. This makes the body perceive low estrogen, removing the negative feedback, causing increased GnRH, FSH, and LH secretion, which stimulate follicular development and triggers a stronger, more consistent LH surge. For more resistant cases, injectable gonadotropin containing FSH and LH can directly stimulate the ovaries. Ovarian response is monitored via ultrasound to ensure a mature follicle develops. This approach is called controlled ovarian stimulation. Intervention 2. Lifestyle by stress management and healthy body weight. The second intervention is lifetime modification. Chronic stress elevates cortisol level, which directly suppresses GnRH pulsatility in the hypothalamus, reducing FSH and LH output. Stress reduction through adequate sleep, moderate exercise, and mindfulness can help restore normal GnRH secretion. Additionally, both underweight and overweight states alter estrogen metabolism and insulin sensitivity, further disrupting the hormonal axis. Achieving a healthy BMI supports more consistent and stronger LH surge, improving cycle regularity and ovulation frequency. If natural conception remains unsuccessful, after intervention, assisted reproductive technology offers two primary options for this couple. Option 1. Intra-uterine insemination, IUI. Intra-uterine insemination, IUI. This involves combining ovulation induction with direct placement of concentrated washed sperm into the uterus around the time of induced ovulation. This bypasses cervical barriers and improves the chance of sperm reaching the fallopian when the oocyte is released. Option 2. In vitro fertilization, IVF. If IUI fails, IVF involves stimulating the ovaries to retrieve multiple mature oocytes, fertilizing them with the husband's sperm in laboratory, and transferring the resulting embryo into the uterus. IVF completely bypasses the timing problem, give full control over fertilization, and significantly increase the probability of successful pregnancy in women with ovulatory dysfunction. Physiological outcome if fertilization occurs but implantation fails. If fertilization does occur but the zygote fails to implant, the following happen. Normally, a successful implanted embryo secretes HCG, which is human chorionic gonadotropin, which signals the corpus luteum to keep producing progesterone, which maintaining the endometrium. Without implantation, no HCG is produced. The corpus luteum receives no rescue signal and degenerate, which is a process called luteolysis. Progesterone as estrogen level falls sharply. The stratum functionalis, which is the functional layer of the endometrium, loses its hormonal support and shape. The woman experiences what happens to be normal menstrual period, around 14 days after ovulation, through a very early fertilization event, has technically occurred and failed. This is known as biochemical pregnancy loss. To summarize, the patient's reduced LH surge disrupt the hormonal cascade required for reliable timely ovulation, making successful natural fertilization unlikely. The narrow viability window of both oocyte and the sperm means that even occasionally ovulation may not align with intercourse timing. Medical interventions such as clomiphene or gonadotropin therapy combined with lifestyle modification can restore ovulatory function. If it is insufficient, IUI and IVF provide effective ART solution. Understanding the biology behind ovulation and fertilization is fundamental to diagnosing and treating fertility. Thank you for watching.